DNA damages. DNA damages of two types, exogenous damage and endogenous damage. The name indicates like exogenous is from the external factors and endogenous damages are within the body. Normally during a metabolism, that is which is taking place in our body, produces reactive oxygen species. This in turn will have some effect on your DNA. This is known as endogenous damages. One of the best example is deamination. When you go for exogenous damages, the best example is UV radiation, gamma rays, X-rays, which come in contact, which lead to the DNA damage. So today you're going to see in detail what is DNA damages and how we're going to repair the DNA damages. It's not we that the body is going to repair itself these DNA damages. So first is DNA damages. The DNA damages is broadly divided into, first one is the exogenous damages and the second one is the endogenous damages. So what are these exogenous damages? The best example will be the ultraviolet rays. So the examples are ultraviolet rays can be either A or B type, thermal disruption, or hydrolysis reactions. It can be also either due to the biological factors like viruses. Now moving on to the endogenous damages, it's mainly because of the metabolic process, leading to the production of reactive oxygen species, which is in turn gonna affect your DNA. So now we are going to see these things in detail. The first one is, if you take on the endogenous damages, so you all know what is DNA made of. This is nothing but nucleotides and you have a sugar backbone with the phosphate molecule. So in the nucleotides you have the four main type of nucleotides for DNA is adenine, guanine, cytosine and thymine. If any one of this nucleotide is mismatched, that means if instead of a cytosine pairing with a guanine, the cytosine is not pairing with the guanine and it is gonna mismatch with the thymine, or there is a missing of a pair between the nucleotides or the sequence, this will lead to this damage. And the next is forming a dimer formation, that is when instead of pairing with cytosine and guanine, cytosine pairing with the thymine molecule, creating a cytosine thymine dimer. So this also will lead to a DNA damage. The other few examples for DNA damages will be hydrolysis reaction, oxidation, deamination, etc. Oxidation is nothing but addition of an oxygen molecule to your nucleotide. So where is a base pair or where is structure of the molecule itself will change, leading to a DNA damage. So these things gets corrected before the replication process or via replication process. So the first endogenous damage we're going to look into is oxidation of bases. The bases are nothing but oxidation of bases. The bases are nothing but the nucleotides. So one examples I'm going to give you is oxidation of bases. You can base the example is 8-oxo 7,8 dihydroguanine. So this is addition of an oxygen molecule to a guanine molecule, leading to the oxidation of the bases. There is a DNA damage. The next type is alkylation. So the name indicates addition of an alkali. So usually best example which for alkylation is methylation. Under methylation, one example can be given is 7-methylguanine. So addition of a methyl group to your nucleotide sequence. The third one is hydrolysis. Hydrolysis of bases. Examples are deamination. Even you can take depurination, depyrimidation, You all know what is purine and pyrimidine molecule. Under purine you will have adenine and guanine. Under pyrimidine you will have cytosine and thymine. So removal of any one of this nucleotides from your bases will lead to depurination or depyrimidination, or the hydrolysis reaction takes place where the bond which is been between AG or C and G or A and T is been reversed. The last one is bulky adder formation. That is any of the two. For example, you can take benzyl A-pyrin, alpha-pyrin, diol epoxide guanine. So you can see a long structure of a group or a chemical group attached to your base, so leading to bulky adder formation. And the next one is mono adder formation. The base example for mono adder formation is change in single nitrogenous bases. For example, if you are having a cytosine which is gonna pair with your guanine and adenine is gonna pair with a thymine, if one of the nucleotide base has been replaced or if it is removed, it will lead to mono adder formation. And similar way you can also take diaded formation. Diaded formation where Cg also pairs with the T molecule. So there is a two nucleotides, that's why it is termed as dinucleotides, and the A remains empty. So this is known as diaded formation. So these are the simple type of endogenous damages which is occurring in your body. So broadly DNA damages are divided into two, exogenous and endogenous. Under endogenous you will have oxidation, alkylation, hydrolysis, bulky adder formation, mono adder formation and diaded formation. Now we are going to move on to the next thing. It's known as exogenous damage. It usually occurs due to the external factors like UV light, X-rays, gamma rays, or if you come under a variable heat formation, that is thermal disruption. This will be leading to your DNA damages. So we're going to see one by one. The first one is UVA light. It is responsible for the formation of cytosine and thymine dimers. So you can see cytosine and thymine. Instead of cytosine pairing with a guanine and thymine pairing with an adenine, they two themselves is pairing to form a dimer. It is mainly because of a UVA light. Next you have UVB light. Exogenous damages, it's of two types, UVB and UVA. So UVB light, cytosine and thymine dimers will be formed. Under UVA light, the free radicals will be generated. These free radicals are responsible for DNA damage and it is called indirect DNA damage. So this UVB light is gonna pair, that is cytosine and thymine dimers are coming together, and you all know cytosine and thymine will come under what? The cytosine dimer comes under pyrimidine. So cytosine and thymines are forming nothing but pyrimidine dimers. So this pyrimidine dimer formation which is direct because of the UVB light is known as direct DNA damage. And UVA light which is forming the free radicals and it is known as indirect DNA damage. It is gonna create a metabolic process where free reactive oxygen species will be produced and it is in turn gonna affect your DNA and is known as indirect DNA damage. Next, third one is thermal disruption. Like after a specific temperature, like for example 80 degrees Celsius temperature, your body cannot withstand a heat. In that process, first the DNA mutation or there is a process where missing of the bases will take place. So thermal disruption is also a responsible factor for your DNA damage. There are some bacteria which can withstand this temperature, and they are known as thermophilic bacteria. Example is Thermus aquaticus. So first we have seen the two types of damages, endogenous damages and exogenous damages. Endogenous damages are usually seen because of the metabolism which is happening in our body, which is going to produce reactive oxygen species, which is going to affect in turn. So there are different types of process taking place in an endogenous damage. For example, oxidation, alkylation, deamination, hydrolysis, and diaded formation, monoaded formation, single aded formation. So it is all between the bases. If a base goes missing, or if base is attached to a chemical compound which is produced in a body, that is produced from a reactive oxygen species, will lead to an endogenous damage. When you move on to an exogenous damage, exo means external factors. The factors can be anything like UV light, thermal disruption. Under UV light also you have A form, B form, and certain infrared rays, X-rays, gamma rays, and certain type of viruses also will lead to DNA damages. So far we have seen what is DNA damages. Now we are going to see how we can repair this DNA damages, or how our body is repairing within itself these DNA damages. So the DNA damage repair mechanism. So the first DNA damage repair mechanism we are going to see is direct reversal repair. It is of different types of mechanisms. One is direct reversal repair. The second one is known as excision repair. The third one is known as recombinational repair. And the fourth one is known as SOS repair. So there are different types of repair mechanism which is taking place in our body for upcoming this DNA damages. So the first one is direct reversal method, the second one is the excision repair, and the third one is the recombinational repair, and the fourth one is the SOS repair. So now we are going to see in each mechanism in detail. The first one, direct reversal repair mechanism. Direct reversal repair is divided into two methods, photolyase method and methyltransferase method. So this photolyase and methyltransferase are nothing but enzymes which is catalyzing few reactions in the body, which is also helping the body, helping the DNA to overcome this repairs or overcome the damages. So we are going to see how this has been worked. So the first method is photolyase method, which comes under direct reversal method. So under photolyase method, you have a photolyase enzyme, which is a major factor for this reaction, photolyase enzyme. You usually have studied there are different types of DNA damage. One of them is forming a dimer formation. So usually a dimer is seen between a cysteine and thymine molecule, which is closely nearby, and it then is gonna bind together to form a dimer, which is known as pyrimidine dimer. Now removal of this pyrimidine dimer, how our body is acting. So the photolyase enzyme is gonna remove the pyrimidine dimer. This pyrimidine dimer is formed when in the presence of an UV light. So you can take the UVA light. I am taking it as an UVA light. The UVB light. The UVB light will be forming a pyrimidine dimer. The pyrimidine dimer which is formed within the DNA is being controlled or edited by photolyase enzyme. This photolyase enzyme is present in the body, but this is getting activated only in the presence of a visible light. So under UV radiation, if a pyrimidine dimer was formed, it is getting reversed or rectified with the presence of a visible light by the photolyase enzyme, where your pyrimidine dimers are cleaved. So this is one of the method for DNA repair. So for the structure, so you have a DNA and you have the nucleotides. For example, you can take this as A pairing with T, C pairing with G, and you can have T pairing with A, and you also can have T and A, and you also, the first here is C. So this dimer is to be rectified, which is formed in a body, and this dimer is formed by UVB light. Right? You can take any of the UV light, and the dimer is formed with the help of this UVB light. Now this dimer has to be rectified. Rectified in the form of visible light. In visible light, the photolyase enzyme, which was within your body, gets activated. So what happens? It is gonna split the nucleotides. So the thymine and the thing, now it is T is gonna pair with A and C is gonna pair with the G molecule. So this is how the process is getting rectified. The second process is excision repair. Excision repair mechanism. So DNA damage repair mechanism are divided into four: direct reversal method, excision repair, recombinational repair, and SOS repair. So far we have seen direct reversal method, photolyase method. Photolyase is nothing but an enzyme which gets activated in the presence of a visible light, and it is gonna cleave the dimer, which is a pyrimidine dimer, which is formed because of a UV light. Right? So the photolyase enzyme will get activated in such a way that the dimers will get splitted up, so the process becomes normal, that is your DNA damage has been rectified. Now second method which comes under direct reversal is methyltransferase method. The second method is methyltransferase method. It involves the involvement of a methyltransferase enzyme, which is also known as alkyl transferase or alkyl transferase enzyme. So methyltransferase is also known as alkyl transferase enzyme. So usually cysteine and adenine, that is cysteine and adenine residues will be found in the nucleotides, that is cysteine and adenine, and this reaction is catalyzed by methyltransferase or DNA methyltransferase enzyme. So DNA methylation normally occurs in your body. DNA methylation normally occurs in cysteine and adenine residues. DNA methylation occurs usually in cysteine and adenine residues. So this process has been catalyzed by DNA methyltransferase or alkyl transferase. So in the presence of an alkylating agent, giving an example, methyl methane sulfonate, this DNA methyltransferase enzyme, instead of catalyzing the cysteine and adenine residues, it is gonna catalyze the guanine residues. So leading to the formation of O6 methyl guanine. So what happens is methyl transferase enzyme is one of the method which is used in direct reversal method. It is playing with the help of an enzyme known as methyl transferase enzyme, which is normally a DNA methylation reaction will take place in our body in cysteine and thymine residues. Cysteine and adenine residues. So methyl transferase enzyme, a methylation or DNA methylation takes place in a body between cysteine and adenine residues. So what happens during a presence of an alkylating agent, that is methyl methane sulfonate, this causes the causes the methylation of the guanine residue. That means methyl group goes and attached to the guanine nucleotide, leading to O6 methyl guanine formation, which is not a proper strategy appearing in a body. So this type of DNA damage is rectified by the methyl transferase enzyme or alkyl transferase enzyme. So this enzyme will find out where the methyl guanine is been present, and it is gonna remove the methyl and the guanine, that is by cleaving this bond. So this methylation reaction takes place between the methyl and guanine, that is demethylation. So far we are studying methylation. So demethylation is removing of the methyl group from the guanine residue. So what happens is now the methyl transferase DNA methyl transferase enzyme, which was catalyzing the reaction between cysteine and adenine residue, now recognizes the O6 methyl. Now it recognizes the O6 methyl guanine and forwards demethylation, that is removal of methyl and guanine. This can be well understood in a structure form. You all know the structure of your O6 methyl guanine, that is in the 6th position you will have. So you have a compound known as O6 methyl guanine. It's been converted into normal guanine. That is active methyl guanine methyl transferase to consumed. This is consumed by the reaction MGMT. So methyl transferase enzyme, it's also known as alkylate transferase enzyme. DNA methylation usually occurs between cysteine and adenine residue. So in the presence of an alkylating agent, we are given example methyl methane sulfonate, which is gonna catalyze the methylation of cysteine and guanine residue instead of adenine. So this guanine will have a methyl group, that is O6 methyl guanine. Now demethylation process, which is gonna rectify the DNA damage. You're gonna use O6 methyl guanine, undergo demethylation where methyl and guanine is been removed, and this process is been catalyzed by DNA methyl transferase, which is gonna recognize this methyl guanine residues in your body, undergo demethylation and remove methyl and guanine residue. So what happens, this active methyl transferase enzyme, which is present in our body, is utilized for the reaction. So we can tell it as consumed methyl guanine methyl transferase enzyme. So I have written methyl guanine methyl transferase enzyme, since it is going to act on the guanine molecule. Next thing is the excision repair. So it is a process where the photolyase enzyme is been not used by our body. Instead, it has been using or bypassing in another method for repairing the DNA damage. So what happens here is, you have a damage. It's for example, you can take it as a dimer formation. There is a pyrimidine dimer formation in your nucleotide sequence of your DNA stretch. So you have a long DNA stretch with the nucleotides, and a few nucleotides are form a dimer. For example, you are taking a pyrimidine dimer that is between cysteine and thymine residue. Now instead of photolyase enzyme coming and correcting the process, we are going to use the endogenous nuclease. That is, endogenous nuclease or the enzyme or endonuclease enzyme is going to cleave the nucleotide, that is 5 to 8 base pairs, apart from your dimers. So what happens? You will have a break of this long stretch of the DNA. Now it has become a small stretch, and in the middle you will have your dimer formation. Now what happens is, once this process has been over, next what happens is, this has been recognized, or this dimer has been removed or recognized by certain genes which is present in your body. Usually this process is been taken by 3 types of genes: UVR, A, B and C. So this UVR, A, B and C are the genes which will recognize there is a damage which has happened in your DNA. So now for example, if there is a pyrimidine formation that is happening between cysteine and thymine residues, this has been recognized or identified by 3 types of gene: UVR, A, B and C. So once this gene is recognized, the endonuclease enzyme comes and cuts the nucleotide, and now a small stretch of DNA with dimer is there. Now the next enzyme UVRD comes and removes the dimer. Now what happens, once the dimer is removed, there will be a small gap between the nucleotides because of the cut. Now this has been ligated by the ligase enzyme. So there are few role of enzymes in this process without using photolyase enzyme. So now we are going to see this method in detail. So excision repair we can tell, the first, it bypasses or it will not use the photolyase enzyme. It will not use the photolyase enzyme for repairing, and it is also variations. This is like cut and patch. You cut and you join. This mechanism can also be said cut and patch. First one is, first we will have the incision step. That is incision, a repair endonuclease enzyme recognize the damage. For example, you can take it as thymine dimer. 2 thymine is combined together to form a thymine dimer. So first you have a long stretch of the DNA, and here so 1, 2, 3, 4, 5, 6, 7 and 8, 9, 10. 1, 2, 3, 4, 5, 6, 7. So the first one is, you have a dimer formation here. The left nucleotides are here. So this is not paired. For example, you are taking as thymine residues. The first one is recognized by endonuclease. So it is going to cut the nucleotides. For example, 5 dash, 3 dash, 3 dash, 5 dash. It is going to cut first 8 nucleotides apart in your 5 dash direction. So 1, 2, 3, 4, 5, 6, 7 and 8. So it is going to make an incision, and here it is going to make 5 nucleotides apart. 1, 2, 3, 4 and 4th and the 5th. So this is the first is recognized by the endonuclease enzyme, and it is going to cleave your nucleotides, as the name indicates. It's a nucleose enzyme. And the next process is, once it has been cleaved, this process is also catalyzed, or it has been recognized by UVRA, UVRB, UVRC genes. So this has to be remembered. Now the dimer has been removed. Now after the removal of dimer, the dimer is removed by UVRD gene, and the space or the gap between the nucleotides, once this gaps between the nucleotides is filled by ligase enzyme. So this is the process of excision repair. Instead of using a photoligase enzyme, the molecule within itself will gonna produce an endonuclease enzyme. It is nothing but a cut and patch mechanism where you have a molecular sister known as endonuclease enzyme. It is gonna cut the DNA where the dimer is being formed, not at the exact dimer formation, but a space little apart. For example, here the process is like in 5th dash position, 8 nucleotides apart, and in the 3 dash direction it will be 5 nucleotides apart. So once it is cut, the process is over. Now you have a small piece of a DNA having a dimer. Next, the gene involved area is UVRD. So the D, you can remember it as a removal of a dimer from the sequence. So the removal of the dimer is carried out by the UVRD. Once the D recognizes the dimer, removes it off, you have a gap which has been formed by the removal of the dimer. And you also have a gap between the nucleotides. So this has been filled by ligase enzyme. Now the third one is the recombinational repair. The name itself indicates the process will undergo a recombination and gets the mistake corrected itself. That means the source or the nitrogen or the DNA which is having a DNA damage will undergo recombination, and during a recombination the damages will be corrected, which is no longer gonna affect the source, that is the daughter DNA which has been produced from the parental DNA. So we're gonna see recombinational repair. Like the first one is the effect of thymine dimer on replication can be studied from this process. The effect of thymine dimer on replication. The second one is in recombinational repair, the gaps which has been formed in the daughter strand and the parental strand. During the dimer removal, there will be a gap formed, is repaired. The third one is the thymine dimer is removed by excision repair. So it's not going to affect your replication process. So you have the involvement of two processes, excision repair as well as the recombinational repair. And this process is usually carried out by a specific protein known as RECK protein. The recombination is dependent on RECK protein, which is the major factor for this RECK protein. Now, once this is involved, the undamaged parental strand recombines into the gap opposite to the dimer. So once dimer is removed, there is a gap. So that will be recombined. And the sixth process is the gap in the undamaged parental strand undergo excision. So recombinational repair, so far we have studied about thymine dimer formation, going direct reversal method, which you have direct reversal under that photolyase and methyl transferase method, and the second method was an excision repair method. Instead of photolyase, you're gonna use the endogenous nuclease enzyme, that is endonuclease enzyme, which is present within your body for correcting, that is cut and patch mechanism. And the third mechanism is the recombinational repair, where your parental source with the dimer will undergo a replication, but it's not no longer gonna affect the doctor's stand, because during the process it is been carefully rectified in such a way that the dimer is not gonna form in your doctor DNA. Now the dimer which is already present in your parental DNA can be removed by the excision repair. This is a previous technique. So here there is a combination of both technique. Replication also is there, the gap and the dimer is also been removed by the excision repair mechanism. So this is, you can take as a DNA. So you all know about the leading strand and the lagging strand synthesis of your strands during the replication process, right? So 5 dash, 3 dash, 5 dash, 3 dash. So leading, lagging. Now recombination is dependent on Rec A protein, that is recombination A protein. So it has identified that there is a damage. So it has been identified that is a damage, that is a dimer. So what happens is, the damage, you have this gap formation in your daughter nucleotide, this is a parental nucleotide, and there is also a gap formed in the lagging strand synthesis because it is nearly opposite. When the dimer is been removed off, or the with the presence of the dimer, there is a gap formed between this and this. Now if you see, so this is a recombined parental DNA, undamaged parental DNA which is gone undergo excision repair. So you will have undamaged replicated DNA. So you can see a dimer is formed here, it is undergoing replication, and by the excision repair this damage has been rectified and you have undamaged replicated DNA. So the last method is the SOS response method. The SOS response method of DNA repair mechanism. So fourth method is the SOS response method. The SOS response method, like normally we have seen the direct reversal method, excision repair method, recombinational repair method. When you see, you are not gonna deeply look into the genes, you know overall what is the process controlling the reaction. Now for when you take into the SOS response method, the DNA damages when appears, there will be certain things or certain proteins which will be released, which is indirectly gonna repair those damages. So you have an SOS response. Here you gonna look out at the small out rule of a operon. So where the operon is nothing but you have a one single promoter, you have an operator, you have a large amount of structural genes. So this genes is gonna control the function or produce a byproduct. So this is how the process takes place. So now we're gonna see what is SOS response in detail. So greater than, if you take the SOS gene, the SOS gene is controlled by a repressor or a molecule known as LETS-A. So LETS-A is the first highlighter here, the LETS-A protein. It also has a two sites, activator and a repressor. So you have an activator and a repressor site in this LETS-A protein. The repressor molecule is been activated, whereas your activator molecule is suppressed. So your repressor molecule is activated, which is going to repress your SOS gene. So this SOS gene, for example, it is given as UMUCD. UMU, C and D are the genes which are the SOS genes which can repair the DNA damages by itself. but the let's A protein or the repressor molecule is suppressing the SOS gene, so it no longer can repair the DNA damage. So what we are gonna see is how this let's A will be released. So if for the previous process we have studied a Rec-A protein, right? So during a DNA damage, before undergoing a replication, a DNA damages is being recognized by a Rec-A protein. So this Rec-A protein, what happens is, this Rec-A protein will know there is a damage which is present in the DNA. So it will be produced in more amount so that there will be a recombination process, that is after a replication, this recombination process like excision repair can take place on the dimer. So this is the role of Rec-A protein. So when Rec-A protein is produced during a damage, so Rec-A retin is produced. So Rec-A protein is produced during a damage. So what happens? This Rec-A protein, when increased in the concentration, it goes and affect, or if it goes and act on the let's say. So what happens is it gets activated. So if it is getting activated, your SOS gene gets activated. Thus, your DNA damages will be repaired. So it will undergo recombination, excision repair. But the only thing is the genes involved here are U, M, U, C, D, right? So you have let's say gene or let's say protein which have an activator and a repressor site. The repressor site is been hindered and the activator site is moved, so that it is going to suppress the SOS gene. The SOS genes are like U, M, U, C and D, which is going to act against the damages which is present in the DNA, normally which you have seen in recombinational process. So what happens is during a DNA damage, the damage has been identified by a Rec-A protein. So this Rec-A protein goes and act on the let's say protein's activator site. So what happens? The let's say site no longer gets activated. It can no longer stay in the or control the SOS gene. So the let's say protein will be released out. This SOS genes will control the DNA damages by activating the U, M, U, C and D. So the Rec-A protein will recognize, it will undergo replication and the dimer will be removed by the excision repair mechanism. So this is the overall process of DNA repair mechanism. So DNA damages like endogenous and exogenous damages are repaired by our body by a direct reversal mechanism, SOS response, recombinational repair mechanism and excision repair mechanism.